My name is Josephine Bwogi, and I'm glad to present to you my PhD research work, which is entitled Rotaviruses in Uganda, Molecular Epidemiology and Potential Zoonotic Transmission. That is the outline of the presentation which I'm going to make. We have two publications have come out of this work, and one is still a manuscript which is yet to be submitted. For this presentation, we have some definitions we are going to follow. We have the anthroponotic, which means the spread of disease from humans to animals. Genome, which means complete set of organisms' genetic material. In this case, we are going to refer to RNA because rotavirus is an RNA virus. And then we have phylogenetic analysis, which is a method to identify the evolutionary history or relationships among organisms, or DNA. And sequencing is the determination of the order of the nucleotides in DNA or RNA. Zoonotic, referring to transmission of diseases from animals to human. Globally, diarrhea has been reported to be the fourth cause of deaths in children under 5 years, with rotavirus being the most common cause of death. In Uganda, the prevalence of rotavirus has been reported to be 39 to 45%, and the prevalence in animals has ranged from 1.2 to 65% globally. And the prevalence has varied depending on the animal species, the age at which the animal has been examined, and also whether the animal had symptoms or not. This map shows us the deaths of children under 5 years who are reported in 2013. If you notice that globally, more than 56% of deaths have been reported in Africa and Asia, with Uganda here reporting about 105,000 deaths. The rotavirus is a wheel-like structure, and it has 11 segments of double-stranded RNA. These 11 segments, each of them encodes for a protein, that is VP1 to VP7, NSP1 to NSP4, and then segment 11 codes for two proteins, that is NSP5 to NSP6. The rotavirus is made up of three layers. The middle layer, what you need to notice is the middle layer, which is the VP6, is used in the classification of rotaviruses. And so far they've described groups A to J, and the most common cause of infections is group A, and that is what we have studied in this study. And also to note is the outer layer, which these spikes are VP4, and this is VP7. These are used in the classification of rotaviruses. This classification of rotaviruses into using that VP7 and VP4, that outer layer, classify them into either G-type or P-type, and the circulation varies geographically and also in time. This distribution is also species-specific. What do I mean? If you look at the G-types, for example, in the humans, the G-types, the most common G-types are 1, 2, 3, 4, and 9, and 12. And these ones which are in samples, these ones are rarely seen, and these ones are not seen. And yet you find that some of these genes which are not seen in humans are present, for example, in other species. Like, for example, the also or the P. Also, likewise, for P-type, we find that there are some P-types which are present. The ones which are just shows that the ones which are most common. And you find that you can have, for example, this G3 is common it's rarely found in humans, but in pigs it's not found at all. Another way we classify rotaviruses is by using the nucleotide sequence identity of the rotavirus genome. So we have some cutoff values. When you get a certain cutoff of how similar those nucleotides are, then it gives you that genotype. You're able to name that genotype. And this classification, we use it in studying rotavirus evolution and interspecies transmission. Rotaviruses, the main route of transmission of rotaviruses is orophycal, and it can be transmitted from animals to humans that don't have. It can be either transmitted directly or indirectly from contaminated objects and hands. Sometimes we get transmission of rotaviruses from people who are not infected or animals that don't have symptoms. Then they transmit to other people, although they don't have symptoms. And sometimes we have transmission from one species to another. And when we have transmission from one species to another, we end up having rotaviruses. Sometimes we have strains, we end up having new strains of rotaviruses coming up. And then we study this interspecies transmission, we use complete genome sequencing. Some risk factors of rotaviruses have been described. Rotavirus infection has been reported to occur early in children, to occur early in earlier years in the children in the low-income countries like ours. And also, most of the infections, we need a lot of infections to get immunity in our settings, to be able to get immunity. And also, breastfeeding has been reported to be protective to direct diseases including rotavirus, although this one is controversial. Some studies have shown that breastfeeding is not protective, other studies have shown that it is protective. Then also rotaviruses have been reported to be common in children attending daycare, and also when you have contact with children who have been exposed to rotavirus infection. As for HIV, studies that have been carried out so far, they show that there is no increased risk to rotavirus infection when you have HIV infection. Also, the rate of the disease doesn't increase when you have rotavirus infection. What led us to do this study? Diarrhea is the top three cause of death in the children under 5 years. And as a strategy to control this rotavirus diarrhea, WHO proposed a certain number of strategies. And one of the strategies which was proposed was rotavirus vaccination. Uganda has embraced this. In 2018, we've introduced rotavirus vaccination. But however, rotavirus vaccine efficacy has been reported to be lower in developing countries like ours than in the other developed countries. And they think one of the causes of that low vaccine efficacy might be due to the high variety of rotavirus strains. And one of the things that has been hypothesized that causes these high rotavirus vaccine strains is interspecies transmission of rotaviruses. And to notice that Uganda has a high population of domestic animals, and we live closely together with these animals. And we think this close proximity with the animals can lead to interspecies transmission of rotaviruses. And to notice that rotavirus transmission has not yet been described in Uganda. And we also have limited data on rotavirus prevalence and risk factors. So the information on rotavirus prevalence, risk factors, and transmissions is required by our Ministry of Health to be able to inform their diarrhea and rotavirus control programs. As I already mentioned, Uganda introduced rotavirus vaccination in 2018, and also globally. And I've also mentioned that we need to understand, and few studies have been carried out in Uganda, prevalence and rotavirus strains, risk factors, and no studies have been carried out on transmission, which we need for the RP control strategies. And also to notice that globally, few studies have been carried out on co-surveillance of humans and animals using whole genome sequencing. These studies would really help us to understand these transmission problems. The studies that have been carried out so far are on using nucleotide sequences, which don't give you a complete picture of how bad this problem is of interspecies transmission of rotaviruses. So we are going to undertake this study so that we can be able to get baseline information. We took the study to be able to get baseline information pre-rotavirus vaccination so that we can provide information on rotavirus prevalence, strains, transmission, and risk factors, which will be used by our EPI program to assess the impact of the introduction of rotavirus vaccination introduction. And we also contribute to the global knowledge of interspecies transmission of rotaviruses. What research questions did we look at? We looked at we asked that what is the prevalence of rotavirus infection in under 5 children in Uganda? What are the characteristics of rotavirus infection in under 5 children in Uganda? What is the prevalence of rotavirus infection in domestic animals in Uganda? And what are the characteristics of rotavirus infection and also of the animals that got this rotavirus infection in Uganda? What are the genotypes and sequences of the rotaviruses? And wanted to see whether they were similar. And what is the prevalence of rotavirus in the domestic animals? And also wanted to study the characteristics of infection of the people who are in contact with these domestic animals. So the hypotheses which we set out to do were domestic animals contribute to rotavirus diarrhea in children less than 5 years old in Uganda, and that asymptomatic household contacts act as reservoirs rotavirus infection of children less than 5 years old. So our major aim of the study was to determine rotavirus prevalence, strains, risk factors in the under 5s with acute diarrhea, and to describe interspecies transmission of rotaviruses in Uganda. So we looked at, that was our conceptual framework. And the conceptual framework was showing us the relationship. The conceptual framework was showing us the relationship between acute rotaviruses and the factors which can lead to acute rotaviruses in children. We looked at possible causes of rotaviruses. There were socioeconomic factors, presence of environment, presence of contaminated environment. We thought also clinical factors may affect the way acute rotavirus may present. I've already mentioned that breastfeeding may be protective to rotavirus infection. And then we thought that there might be some transmission of rotaviruses from asymptomatic household contacts to these ones, and also transmission from the animals to these. So these ones inform some of the studies which we carried out. So this PhD work was composed of 3 studies. So study 1 has already been published, was published in Archives of Virology. And the specific objectives for study 1 were to determine the prevalence and strains of rotavirus in under 5 children hospitalized with acute diarrhea in Uganda, to describe the factors associated with rotavirus infection in under 5 children hospitalized with acute diarrhea in Uganda, to determine the prevalence and associated factors with rotavirus infection in the household contacts of the under 5s hospitalized with acute diarrhea, and to determine the prevalence of rotavirus infection in the domestic animals that were staying in these households of these children who were admitted with acute diarrhea. So this table summarizes the methods we used. This was a cross-sectional study which took place between September 2012 and September 2013. And it took place in 4 hospitals, 2 hospitals in Kampala, that was Mulago, and Sambia Hospital, and 2 hospitals from Masaka and Chitovu. We selected Mulago Hospital because currently there was already surveillance rotavirus which was ongoing, so we said, and it receives a number of patients, so we said it because we shall be able to obtain our sample size. And then Masaka hospitals were selected because we have had reports that 2008 census of animals reported a high population of pigs in Masaka district. And since we're trying to study zoonotic transmission, that is why we included Masaka. The included Sambia Hospital. These are private hospitals, and we thought this one should be included since, because maybe the patients may be different from the ones that are attending the government hospitals, which is Masaka and Mulago. So the patients we looked at were aged 2 to 59 months old, who had a diarrhea that was less than 14 days. It was not bloody. Then we also looked at the household contacts of the children who were rotavirus positives and the animals in those households. And we purposefully selected the hospitals, as I already mentioned, and for the children, we only enrolled them consecutively as they came into the hospital. And for the household contacts, so after enrolling the children, those children who tested positive went back to their homes and recruited the households and the animals in those households. So to determine the sample size that will be used for this study, for the prevalence studies, the prevalence of the rotavirus in the household contacts and the children, we use the Kish-List formula, and then we use the Chessman formula to get the sample size for the risk factors which we studied in the children. And the sample size in the hospitals, to find out how many we recruit from each hospital, we use probability sampling for the hospital, using the prevalence of rotavirus diarrheas which were reported in 2011. And those are the demographic. These are the independent and the dependent and the independent variables we used. We used a structured questionnaire to collect the data on both the animals and the household, all the population, both the animals, the adults, the household contacts, and the children. And then we took off blood samples. From the children to test for malaria and HIV, and stool samples from the children and the household contacts to look for rotavirus. And we tested for rotavirus using rotavirus ELISA in the children, and for the household contacts we use our NSP3 real-time PCR, and also the animals we used ELISA. Data management, we did bivariate, bivariate, and bivariate analysis. For bivariate analysis, we used chi-square and odds ratio to look for associations of the impeded factors with the outcome, which was rotavirus infection. And we also did multivariable analysis using generalized linear models to determine whether the factors were associated with rotavirus infection. And for quality control and ethics, this study was approved by a number of ethics and research ethics committees. The case definitions we used were, we looked at those children who met the criteria and were positive. These ones were taken as confirmed as rotavirus. And then for the symptomatic household contacts, if the adult or child had no disobey the last 21 days and positive, then we took that one to have rotavirus infection. So what results did we get? We recruited a total of 712 children, and they were aged 2 to 54 months. The median age was 10 months. And we found that the prevalence of rotavirus was 37%. And this rotavirus occurred throughout the whole year. This is in the children was placed. It occurred the whole year. It ranged from 25 to 45%, but we saw it occurring the whole year around almost. There was no seasonality observed. Then among the people we recruited, most of the children were above the age of 39, most of them were female, and most of them presented with a very scary score of greater than 9 or greater than 11. This very scary score is a score which is used in rotavirus studies to describe the severity of the disease. And 32.2% of the children were underweight, 34% were stunted, and 175% were wasted. Sorry, 24.6% were wasted. But HIV infection occurred in 4.9% was present in 4.9% of the children. In 4.9% of the children, and malaria was present in 4.7% of the children. We found that there was an association between the stay in hospital and rotavirus, being rotavirus positive. And also we found that we found only 6.7% of the patients had animals were drinking from, we found where the animals were drinking from the same water source as the patients. So what we found interesting, we found, G9P8 and G12P4 were the most the most prevalent genotypes which are circulating in the rotaviruses in the children, and we found a high prevalence of mixtures, but this D12 and D4 was not previously reported in the previous studies. So among the factors which I looked at which were associated, on multivariable analysis, I found that, to notice that, we found that the children who are recruited from Masaka were more likely than the ones that were recruited from paratoid rotavirus infection. The children who are above 2 years were less likely to have rotavirus infection. The children who are wasted were also less likely to have rotavirus infection. And the caretakers who were chance with soap after toilet, their children were protected against rotavirus infection. And also a hospital stay of more than 5 days, then you're less likely to have had rotavirus infection. In the households, a total of 254 household contacts were recruited, and only 4% of these reported symptoms, and all of them tested negative. And the animals recruited 40 animals, and only one had history of diarrhea in the previous 2 weeks, and others two tested negative. So for this study 1, the prevalence of rotavirus which we saw was similar to what has been reported in other countries that have not introduced rotavirus vaccination. So, and also what we noticed that there was lack of seasonality in the rotavirus infection in Uganda. This lack of seasonality may be because of our location. Our location is on the equator, so maybe that those temperature changes. Maybe that is why we don't have seasonality. But also it has been reported that tropical countries where we have high birth rates, and we have high birth rates, like for example what we are seeing in Uganda, usually have no seasonality seen, and you have high transmission rates. Also, what we noticed that most of our children were below 2 years old, and this has been explained by the fact that what has been observed in the low-income countries, most of the children get their rotavirus infection early in life, and then they build immunity. So that is why you may find that since we're looking at hospitalized cases, that's why you see that most above 2 years, we had this children above 2 years that had rotavirus infection. Then we saw that in our wasted children, there was less likelihood of having rotavirus infection. This may be explained that in malnutrition you get atrophy of the villi, and when you have atrophy of the villi, then you cannot have rotavirus entering and replicating. Therefore, you may end up. That's why I think we had no decreased, less likely to have rotavirus in our wasted children. And this imaging of G12P4 which we saw is something which we explain by the fact that we usually get this seasonality change of rotavirus G and P types, which may be a method for the rotavirus to survive in an environment. Then we saw a lot of mixtures of rotavirus genotypes, where I said there were 32%, and we also had some unusual genotypes which were like G10 and P10, and we think this may be suggestive of animal origin, which required further investigation. So in conclusion, about 4 out of every 10 under 5 children hospitalized with acute diarrhea in central Uganda had rotavirus infection. The most prevalent strains causing diarrhea in under 5s were G9P8 and G12P4. The children who are wasted were less likely to have rotavirus infection compared to the ones who are not wasted. And the children, and hand washing with soap after use of the toilet protected the children against rotavirus infection. And we saw no rotavirus infection in our asymptomatic household contacts. So for study 2, this one is still in manuscript form, it has not yet been published. The specific objectives for this study were to determine the prevalence, strains and factors associated with rotavirus infection in domestic animals in Masaka district, to determine the prevalence, strains and factors associated with rotavirus in humans in contact with animals in Masaka district, and wanted to compare the rotavirus genotypes in the animals and humans in Masaka, because because they're staying in the same geographical region. So this was also a cross-sectional study. It took place in December 2013 and January 2014. It took place in Masaka district, and we studied goats, cats, and pigs, and we targeted younger animals. Although, younger animals less than 2 months in the household, but we would also recruit those which are older. And then we recruited the humans that were in contact with these domestic animals in these households. Then we sampled percussively 2 to 4 parishes in each of the 6 sub-counties of Masaka district we studied. And we moved from household to the next, recruiting the animals and the humans, and recruited a maximum of 5 animals in a household. If we found more than 5 animals of a particular species in that household, we did random sampling. Likewise, we recruited a maximum of 5 people from the household. To determine the sample size of what we're going to study, we used Kish-Leslie formula for the prevalence studies. Our outcome variable was rotavirus infection, and we had demographic and socio-demographic variables which we studied. We used a structured questionnaire, and we took off 2 samples from the animals and animal contacts, and we tested for rotavirus on the animals using rotavirus ELISA test and MSP3 real-time PCR to test for rotavirus in the household contacts. We did RV genotyping using listed PCR, and then we did Sanger sequencing on one of the human samples because we had failed to get the genotype using the listed PCR, so we did Sanger sequencing to find out the genotype. Likewise, we did invariant and bivariate analysis on the samples, on the population we studied, but for the animals, to understand the risk factors that we found at bivariate level, all the factors that were less than 0.2, which were less than 0. Which had a p-value of 0.2 or less, or were biologically traceable, we take them in our multivariable analysis, where we use the mixed effect logistic regression, and we consider clustering at household and sub-county level. This study was also approved by various ethical committees. So in this population of animals, we had a total of 1,137 animals, and of these, 281 were cattle, 418 goats, and 438 pigs. And most households reared 1 to 2 animals. Most of the animals were female, 60.9%. And most of the animals were, only 24.3 of the animals were suckling, and only 10.3 animals had history of diarrhea in the previous 2 weeks before recruitment. The proportion of rotavirus positivity was higher in the cattle compared to the rotavirus prevalence in the goats and pigs. This was a significant difference in the proportions. Also, we found a significant difference of rotavirus infection in the different sub-counties we studied. And this is the Fisher's exact value. So the prevalence in the animals, I've already mentioned, was 7.5% in cattle, 2.2% in goats, and 3.4% in pigs, and the overall prevalence was 4% in the whole population. On genotyping, we found many genotypes. There are 5 different G types, G3, G8, G9, G10, and G12 in the animal samples, and we found 6 different P types. That's P4, P6, P8, P9, P10, P11. This is the animals. And what we noted is that the genotypes G3, G9, G12, P6, P8, which were recorded in the animal samples, were similar to what we had found in study 1. So this one shows us the distribution of the G and P types. The other thing to notice that we had a lot of mixtures. About 25% of the samples had mixtures of G and P genotypes, and we failed to identify the G and P in 9 samples. And that was in about 22%, this is 22% of the samples. On multivariate analysis, when we looked at the factors which we studied as risk factors, we found that the only factor that we found significant was the age of the animals. We found that age group, all these age groups which are above 12 to 18, 6 to 12 months, and 2 to 6 months. So the age groups, these three age groups above, which were more than 2 months, when you compare it to the age groups 0 to 2 months, they were less likely to have rotavirus diarrhea. In the household contacts, we recruited a total of 258 household contacts, and they range from the age of 2 months to 67 years. And out of this, only 9.5% of these 258 were children who are below 2 years old. And also, only 12.3% had history of diarrhea, those household contacts. And what we noted is that only 2 of them turned out positive for rotavirus infection. And those 2 ones I'm describing here, one of them was a male child who had history of diarrhea in the previous, who had history of diarrhea. And in the household where this child stayed, there were 3 domestic animals, but all these animals tested negative. And also, they reported that this participant plays with animals. And when we did the genotype of the rotavirus infection which was found in this child, we found it was genotype P4. We were not able to get the G type. Then the second child did not have symptoms of diarrhea. And this child also, there were pigs and goats in that house where this child stayed. And when we tried to type, we failed to get the genotype. So that's why we went ahead and did Sanger sequencing, and we found that they had G98. So what in this study to notice that we found a low prevalence of rotavirus infection, and the reason for this may be explained by the few number of animals that were found in this household. Because remember we said each household had 1 to 2 animals, so that limits the spread of rotavirus infection. And also most of the animals we sampled did not have symptoms. Although some studies in East Africa where they studied asymptomatic animals in pigs, they found a high prevalence of rotavirus infection. The difference between the results and our results would be explained by the fact that in these other pigs, they had a high number of pigs, up to 10 animals, so maybe that's what increased the rotavirus transmission. And they also used different methods of testing. We used ELISA, they used PCR, so maybe that's why they were able to pick maybe more positivity. And we had a difference of prevalence of rotavirus in our different sub-counties. This one requires further studies for us to know what are the different animal husbandries in our different sub-counties, which may be affecting the prevalence of rotavirus which we are seeing in the different households. And also to notice that some of the genotypes found in the animals are similar to those in the humans. This would be an indication that we have interspecies transmission in our setting. And this would require further research by maybe carrying out sequencing on these samples. So in conclusion, the prevalence of rotavirus in symptomatic and asymptomatic domestic animals was low. The P and G genotypes in our animals were diverse. In Uganda, some of the rotavirus genotypes are similar to what has been detected in humans, and the animals older than 2 months were less likely to have rotavirus infection compared to the ones which are aged 0 to 2 months. So study 3. Study 3 has also been published in PLOS ONE. And the objectives of study 3 were to describe the genetic composition of the rotaviruses identified by humans and animals which are residing in the same geographical region, to compare the sequences of the rotaviruses identified in humans and animals of this same geographical region, and to determine the possible origin and evolution of the rotaviruses identified in the humans and animals in these regions who are staying in the same geographical region. So in this sample, this study was a cross-sectional study which took place September 2012 to January 2014. And because in this sample we use samples collected in study 1, the human samples came from study 1 and the animal samples came from study 2. So the study settings are the same as study 1 and study 2. So we used 18 human samples. These were 18 human positives, which are selected out of the 18 samples. And we selected them based on the ones that had unusual genotypes, or which had genotypes which are similar to animals, or also when we tested had adequate viral material to be able to carry out sequencing. Also the animals were collected from we also selected this from the positive samples of study 2. And we only selected 6 because these are the only ones where we were able to carry out sequencing. So we recruited 1 bovine, that's a cow, 1 caprine, a goat, and 4 porcine, those are pigs. And for this study, I won't go through this data because this is what we did in study 2 and 1. And I will explain this further. This study was approved by the different committees, ethical committees. So the laboratory methods we used, we did on the stool samples from the humans and the animals, we extracted RNA, and we used the triso LS reagent to extract double-stranded RNA from the human samples, and we used the Booms method to extract double-stranded RNA from the animal samples. After that, we added lithium chloride so that we could be able to remove the single-stranded RNAs, and after that, we purified the RNAs, which we got, the double-stranded RNAs. We oligogated those double-stranded RNAs, and then we did cDNA synthesis, and we carried out PCR amplification. Following this, we barcoded the samples. This is now we're preparing the samples for sequencing. So we did barcoding of those samples. We had some samples where we found double-stranded RNA where the concentrations was 2 nanograms per microliter. Where it was 2 nanograms per microliter or more, we used an extra xtDNA kit to prepare the libraries. And where we had double-stranded RNA which was less than 2 grams per microliter, we used strip-seq kit. And following that, during the library preparation, we sent the samples for sequencing, and the sequencing method which was used was the Luminary high-cell platform. And when we got the sequences, we assembled these sequences. And the method we used de novo and also mapping method using the Genius program to be able to do the nucleotide sequence assembly. And then after getting the sequences, after assembling these sequences, we tried to see each sequence. So each genotype, each sample now had 11 segments. So now we try to find out which genotype is it for each of these segments. So we used the Rotaviruses program. We also did BLAST-N to compare these sequences we had got with the sequences that have been reported in the genotype, in the gene bank. Then also these nucleotide sequences, we try to see how similar are they to what is in the gene bank and also to each other. So we used the bioedit program. Then we did phylogenetic analysis using maximum likelihood method, and this was mega 6.06 to be able to understand how these nucleotides, the ones we have, how they relate to each other and others which have been reported. So the results of study 3, remember I said we have 11 genomes. So these are the different samples. So for each of these human samples, we found this green refers to one like genome, and then this one, the red, refers to DSM1 like genome constellation. So all the humans and for all the humans, we found only the wild-like and DSN1-like. These are the animal samples. From the animal samples, we find some others which are not wild-like or DS1-like. So when we did that, when we compared our nucleotide sequences, we found that the nucleotide sequences of the human RNAs were similar to each other. Okay? The ones the ones in our study, the 82.2% to 100% identical to each other, and also similar to other rotavirus S strains from other African regions. But then to note is that we found that a few genes, 1 or 2 genes in our human samples was probably of zoonotic origin, because when we compared them to what we found in the gene bank, they were similar. So that is why we thought that probably 1 or 2 genes on the rotavirus strains were possibly of zoonotic origin. And then what was quite interesting, we found that in bovine, this is the cow, all the 11 genes were exactly similar to humans, our rotavirus strains, both the ones in our setting and also a rotavirus strain which has been described in Italy. And then what also we noted that these are the pig strains from the same household. We had 4 pig strains, each two were got from the same household. We found that their genes were similar. For one of them, about 5 of the genes were similar, but for one, all the 11 genes were similar. So we thought that probably that there was some possible transmission, household transmission of rotavirus in the pigs. For the caprin, that is the goat, the caprin strains did not cluster at all with our human strains, except for VP6G, where it clustered with one of our strains from Molabo. And we found that the caprin strains, that is VP1, the caprin nucleotide sequences VP1, VP2, VP3, and NSP5 genes, clustered with other human strains of zoonotic origin. And the NSP2 was clustered with very many other species. So we couldn't even tell which species, because like goats, cows, they were all similar. So we couldn't really distinguish which particular species was most related to it. So this picture is just to explain what I've already said. This is VP1. You find that this is our study sample. So all the blues represent the human samples, and the red represent the animal samples that we studied. So this sample here, this human sample here, is clustering with a goat sample, this is a goat sample from Bangladesh. So this could indicate possible zoonotic transmission. Although these goat samples also had, when we looked at this gene of this goat sample, is a bit similar to humans. So, but this one now we say maybe there's a possibility of zoonotic transmission. This VP6 I've already talked about it. Where I said our sample is clustered with a goat sample here. Also, these samples here, these are human samples from our study. But the samples they cluster with, this one and here, these human samples they cluster with, have been reported to be of zoonotic origin. So probably our gene NSP4 of this sample 183 and 23 may be of possible zoonotic origin. Also here, again this sample, Mulabo 204, is clustering again with a goat sample from Bangladesh. So this sample also, its NSP4 gene may be of zoonotic origin. Again, it's the same information, it's just clarifying the same information. So in discussion, our human rotaviruses are closely related to other human African rotaviruses. And the NSP4, I've already mentioned that the NSP4 of 2 human samples were closely related to that of porcines, of pigs, and also of other human samples which are pig-like, and so they may be of possible zoonotic origin. And also the VP157 was also closely related to human strains from Malawi, which is of zoonotic origin. So also this VP2 from these 2 samples may be of zoonotic origin. Also this VP6, as I showed you, it was closely related to the goat strain, so it may also be a result of interspecies transmission. So when we look at what I've been describing, it just shows you that there's really a challenge. When you do a cross-sectional study like we're doing, it's a bit challenging to identify the exact geographical and temporal origin of this interspecies transmission which I'm describing. And also, one of the pig strains had genes which are closely related to those of humans. So we think that they may have been derived from human to animal transmission. This one is similar to a previous hypothesis. It just confirms a previous hypothesis which has been made by one of the authors, Matt Jensen's, in 2008, which says that pigs and human rotaviruses, which are alike, may have a common origin. So that is why when you do sequencing, you find that they are as nucleic sequences clustered together. So in conclusion, there was evidence of possible zoonotic transmission of rotaviruses in the children admitted with rotavirus diarrhea in our setting. And since we're looking at children who are moderate to severe diarrhea, we think that from our study, we think that most of the rotaviruses seen in the children may be primarily transmitted from person to person, since we had this. So the zoonotic transmission was really scattered. And then we also saw human to animal transmission was identified in our setting. Remember, we had the cow where we got all the genomes are similar to those of the humans, and also the pig which had some bits of genes which are similar to humans. So we identified multiple complex reassortment strains which are derived from complex interspecies transmission of rotaviruses in our setting. So what are the methodological issues we had in our study? So in our study, for the random error, we tried to calculate the sample size. We tried to get the sample size for each of the samples before we did the study to be able to minimize random error. And in calculation, but what we noted is that we felt for study 1, we're not able to get the animal sample size we had set out to study. That is why we could not really get to a conclusion in study 1 on whether animals are transmitting rotaviruses using study 1. And also, in that we selected and for selection bias, how we tried to overcome the selection bias, we had inclusion criteria before we set out the study. So we used this to be able to get our patients. And we also had case definitions. And maybe it's one of the things which had promoted in the random error. For study 1, we had some of for the household contacts, there was a delay to go back of up to 3 weeks. So maybe that delay could have affected our getting the rotavirus positivity. So maybe by the time we went back in 3 weeks' time, we found that is why we found no rotavirus. Probably if we had gone back earlier, we could have got maybe some rotavirus positives. Then information bias, we tried to overcome this by using questionnaires which were structured. We trained our research assistants on the questionnaires, and we've also constantly checked on the information. We reviewed the questionnaires. I reviewed the questionnaires to make sure that the information they're giving is right. Sometimes when the information was not clear, we had to go back to for example, for the hospitals, we would go back to the charts because we're using hospitalized patients, and check that the information they've given us is actually the right information. And for confounding, we looked at this, we addressed confounding at the analysis stage. So in conclusion, I think we had minimal errors in the study, and I think we're able to achieve internal validity for this study. But in terms of generalizability of our study, also, maybe also to mention is that all our studies were cross-sectional studies. So since they were cross-sectional studies, it was not easy, like for example, the risk factors we identified in study 1, you can only say maybe these are associated, but we cannot confirm. And also the zoonotic transmission we saw, which we say we suspect, but we cannot really confirm, because this was a cross-sectional study. And then, since we found some differences in the rotavirus infection between the patients we recruited from Masaka and the ones we recruited from Kampala, we think our results may not be generalizable to a population outside the central region. Maybe we shall need to also carry out other studies in other parts of Uganda. So in summary, the conclusions from this study are: rotavirus infections were common in under 5-year-old hospitalized with children in central Uganda, occurring in 4 out of every 10 children. The most prevalent strains were G9P8 and G12P4. Children above 2 years, wasted children, and those whose caretakers watched times after using the toilet were likely to have rotavirus infection. And we found no rotavirus in the asymptomatic households, suggesting that probably there's limited transmission within households, within the households we studied. And the prevalence of rotavirus in the domestic animals in Uganda was low, it was only 4%. And the animals which are older than 2 months were likely to have rotavirus infection. And the animals which are older than 2 months were unlikely to have rotavirus infection. The G and P distribution in the domestic animals was diverse. There was evidence of possible zoonotic transmission in the rotaviruses in the children, actually with acute diarrhea, with moderate to severe rotavirus infections in the children being mostly due to human-to-human transmission. And also human-to-animal transmission of rotavirus was detected. And we noted that the rotaviruses in the boats and pigs are due to multiple gene reassortment events. So from this study, we would like to make the following recommendations. The Ministry of Health should include ensuring good hand and foot hygiene and messages of diarrhea and rotavirus infection control. It is recommended, now that rotavirus vaccination has been introduced, we should continue with surveillance of rotavirus infection to understand the burden and circulating strains, so that the Ministry of Health will be able to understand the impact. And we should combine the surveillance in both humans and animals to be able to study better the molecular image of rotaviruses. To the community, since rotavirus vaccination has now been introduced, because previously it was expensive and mothers had to pay quite a lot of money, now that it's even cheap, the mother and caretakers should take their children for rotavirus vaccination to decrease that burden of rotavirus diarrhea which we are seeing, and they should ensure good hand and foot hygiene. And since we've seen that there's a possibility of interspecies transmission of rotaviruses in the homes, we'd encourage good animal husbandry in the homes should be practiced to minimize this interspecies transmission of rotaviruses. To the scientists, since we've seen that when you do whole genome sequencing, we get quite some good information on interspecies transmission. I think this should be continued to better understand, to increase our understanding of molecular epidemiology and evolution of rotaviruses. But maybe they should carry out these studies using maybe a prospective study, not the cross-section one we did, so that they can be able to study more samples and be able to understand better this problem of interspecies transmission. And they should also do, the scientists should also do research on animal husbandry and feeding practices to understand what are these factors that lead to interspecies transmission. And maybe, So I would like to acknowledge my supervisors, the doctoral committee. I'd also like to acknowledge my funders. I got funding through Thrive, which was a Welcome Trust grant, and also Cambridge Albedo Research Grant funds. I also would like to acknowledge WHO because some of the tests we did in the hospitals were using kits which were from WHO. So I would like to acknowledge that funding. I would also like to acknowledge the support you got from the Thrive Secretariat, the research assistants. This was a big study, so I worked with so many research assistants, both in data collection and also when we were doing lab testing. I would like to acknowledge the study participants, UVRI, my fellow PhD colleagues, family and friends.